Okay, so we are in the process of discussing the adaptive immune response. Okay, so we've discussed that there are two types of pathogen that you will have to mount responses against. So there are the extracellular pathogens, and we've discussed the complete response that you initiate against them. But there are also these intracellular pathogens. Now, basically, in the case of intracellular pathogens, you have a more complicated response. Okay, so you initiate what I've just described here, which is the activation of these cytotoxic T cells, which will cause apoptosis in cells which are infected with the intracellular pathogen. But you also initiate other responses. Now, let me explain why. Well, basically, it's because of the fact that the intracellular pathogen isn't always intracellular. It can also be extracellular, so it will be intracellular for a good deal of its life, but when it is ready to move from one cell to another, it needs to temporarily go into the extracellular fluid before it can get to its next host cell. Okay, and when it's in the extracellular fluid, it's susceptible to being phagocytosed, and we know if a pathogen is phagocytosed, then its antigens will be presented by these antigen-presenting cells, not on MHC class 1, as was the case when it was actually within the cytoplasm because it had gone there itself, but instead on MHC class 2. Okay, so basically, you can also get antigen-presenting cells presenting antigen fragments of this intracellular pathogen on the surface of MHC class 2, and this will lead to the activation of CD4 positive T cells again. Okay, so similar to the humoral response, basically. Okay, so I also just want to discuss one more thing before we move on to talking about this, which is that it could also be that you have an intracellular pathogen which actually lives within phagosomes. So one reason that you're also going to activate the CD4 positive naive T cell is because you're going to phagocytose the intracellular pathogen whilst it's in the extracellular medium because it's spreading between host cells. But another reason is that there are certain pathogens which can actually live within phagosomes. So they are actually, that as part of their life cycle, they will live within phagosomes. So they are an intracellular pathogen, but they're living within phagosomes. Okay, so here is the nucleus of the cell. Okay, and here is the phagosome. And the microbe will just be living within this phagosome. Okay, so this might be a macrophage, for instance. Okay, and the reason it can live within the phagosome like this is because it's exceptionally good at preventing the lysosomes, which, remember, contain these lysozymes, from fusing with the phagosome and releasing the lysozymes onto it. Okay, so this pathogen is sitting within the phagosome and it's living out its life quite happily and stopping the macrophage from being able to get these lysosomes to fuse with the phagosome and therefore stopping the destruction of itself, basically. Okay, and it will live its life out there and it will divide in there, so it's basically living and taking advantage of the macrophage. Now, examples of bacterial species that are capable of doing this, well, examples of microbes which are capable of doing this, which are bacteria, are mycobacteria species of bacteria. Okay, and the archetypal example of a mycobacteria species is Mycobacterium tuberculosis. So mycobacteria isn't just a single species of bacterium, it's a whole class of species of bacteria. Okay, so a specific species of mycobacteria that's capable of doing this is Mycobacterium tuberculosis, which is the causative agent of TB. Okay, and these bacteria are capable of being phagocytosed inside and going into a phagosome within a macrophage and living within there for incredibly long periods of time. Okay, other bacterial species which are capable of doing it are Listeria species. Okay, and the sort of archetypal example of a Listeria species would be Listeria monocytogenes. Okay, which causes gastroenteritis. So again, these can be phagocytosed. Oh, whoops, what am I doing? Cytogenes. Okay, these can be phagocytosed by macrophages, and then they will live within the phagosome and stop phagolysosomal fusion. Okay, so these are intracellular pathogens. However, they're not within the cytoplasm. They're within a phagocyte. Sorry, they're within a phagosome. So they too will trigger antigen presentation on MHC class 2. Okay, and therefore you will activate CD4 positive naive T cells, not CD8 positive naive T cells. Okay, so when you've activated this CD4 positive naive T cell, which again will have a receptor on its surface which is perfectly matched for this antigen fragment that the antigen presenting cell has presented, okay, what will happen is this cell again will differentiate into a T helper naught cell, abbreviated like so. So T for T cell, the subscript H for helper, and then 0 for naught. Okay, and then these T helper naught cells will divide, so you'll end up with many of these T helper naught cells. Okay, so here's another one. And then what will happen is these T helper naught cells will differentiate. Now in the humoral immune response, they differentiated into T helper 2 cells. However, this is now a cell-mediated response. We don't want antibodies produced. We don't want to recruit B cells, which is what T helper 2 cells are good at. Why? Because they're utterly useless. Why? Because these pathogens spend most of their life intracellularly. Now, of course, it's true that the pathogen has to go into the extracellular fluid to get between cells, and that's why these this distinction is quite brutish, basically. I'm saying that these aren't going to become T helper 2 cells, but in actual fact you'll get a few T helper 2 cells which will recruit a little bit of a B cell response, okay, and those B cells will produce antibodies which may well be able to attack the pathogen in between. But in the main thing that happens in the cell-mediated response is that these T helper 0 cells are going to instead differentiate into T helper 1 cells. So basically most of them will differentiate into T helper 1 cells, some of them will differentiate into T helper 2 cells. In addition, in the humoral response, where we said they all were going to differentiate into T helper 2 cells, again it will be the case that most of them differentiate into T helper 2 cells and some of them differentiate into T helper 1 cells. So really it's a case of favouring one of the two types of T helper cells. Okay, so the reason you don't really want to produce that much antibody is because the antibody just can't get to the pathogen if it's inside a cell. It can only get to the pathogen if it's an extracellular pathogen, because the antibody can easily get out of the bloodstream through these holes that we've opened in the capillary walls and the post-capillary venule walls, and then can bind to the surface of the extracellular pathogen nice and easily. Okay, so what are these T helper 1 cells going to do? Well, basically they're going to produce interferon gamma, okay, which is often abbreviated as IFN gamma. Okay, so this is interferon gamma. Now, interferon gamma is basically a it's a signal to the macrophages that basically says, take no prisoners. It's a signal that gives the macrophages the rights to use brutal force to take down this pathogen, basically. And it's incredibly important for this case where you have intracellular pathogens which are living within phagosomes. The interferon gamma will go to the macrophages which have these intracellular pathogens living within phagosomes, and it will tell them to, come on, just destroy this, basically. Throw a whole new set of tricks at this pathogen that is avoiding phagolysosomal fusion and destroy it. Okay, so that's what interferon gamma does. It tells these macrophages that they can use brutal methods, basically, to just destroy what's within them. It gives them permission to use, to fight fire with fire, basically. Okay, so it's particularly, this little portion of the cell-mediated response is particularly important for intracellular pathogens which are living within phagosomes, basically. Whereas the CD8 positive response up here with the cytotoxic T cells is really important when you've got an intracellular pathogen which is living within the cytoplasm of the cell. Okay, so these are both counted as cell-mediated responses, but in my opinion we could almost do with splitting these into two. This is the one where it's actually intracellular within the cytoplasm, and this is the one where it's more within the phagosome, although of course these distinctions don't hold true. They're not as brutally defined in nature. Instead, you know, this sort of a pathogen where it's in the cytoplasm will lead to both of these, because of course it goes into the extracellular space for a while, and then it will lead to phagocytosis by these antigen presenting cells, and then it will present it on MHC class 2 and activate this pathway. Okay, so that really now is the full picture of the adaptive immune system, or at least it's a picture of the adaptive immune system, whether it's full. So now what we're in a position to do is start to understand what is going to have happened and what's going to trigger the angiogenesis. Okay, so these T cells that we are producing, so for instance T helper 1 cells and T helper 2 cells and cytotoxic T cells, all of these cells that we're producing in massive numbers, they'll be going into the bloodstream, okay? So we want the T helper 1 cells, for instance, to come to our affected area because we want them to secrete interferon gamma onto the macrophages and give the macrophages this right to use brutal tactics, basically. So, in the adaptive immune response, what's going to happen is that the endothelial cells of the capillaries and post-capillary venules are going to start recruiting these T lymphocytes into the interstitial space. So in the affected area, you're going to start getting lymphocytes appearing. You're going to get cytotoxic T lymphocytes, T helper 1 cells, and also T helper 2 cells. Now you can understand very easily why the cytotoxic T cells would want to come into the area, because then they need to go and kill any cells which are infected, basically. So they need to go and meet the cells, basically. In addition, you can understand why the T helper 1 cells would want to come into the affected area, because they want to secrete the interferon gamma nearby the macrophages to give them the rights to kill. But also you do see T helper 2 cells coming. That's a little bit more difficult to understand from what I've told you, basically, because I told you that T helper 2 cells were involved in recruiting the B cells, basically. So if that's their only role, then why on earth do you need them being recruited into the site of infection? What are they going to do? Well, they also secrete all sorts of inflammatory cytokines which are involved there. So basically you get the recruitment of T lymphocytes into the area, and now we're ready to start talking about chronic inflammation and how we're going to induce angiogenesis.